Welcome everyone to the Fujifilm Visual Sonics live webinar series. My name is Sarah Burris. I'm the scientific liaison here at Fujifilm Visual Sonics, and I'm very happy to present a webinar today by Dr. Arvin Soepriatna. He is a postdoctoral fellow at the Center of Biomedical Engineering at Brown University, and he's going to be discussing the characterization of myocardial infarct size using 4D ultrasound. The webinar is being recorded, and the recording of this webinar will be made available later today. All of our participant lines are currently muted and will stay muted during the duration of the webinar. So if you have a question, we ask that you please use the Q&A button throughout the presentation. And that Q&A button is found at the bottom of your screen. Questions that are submitted will be answered at the end of the session, and you can expect the webinar to last anywhere from about 30 to 45 minutes. And it will include a 10 to 15 minute Q&A session at the end. So now what I'd like to do is pass the mic over to Dr. Craig Gorgen, who is an associate professor at the Weldon School of Biomedical Engineering at Purdue University.
Great, thank you, Sarah. Thank you for the introduction. Hi, everybody. As Sarah mentioned, my name is Craig Gorgen. I'm the Leslie A. Geddes Associate Professor of Biomedical Engineering in the Weldon School of Biomedical Engineering here at Purdue University. And I have the privilege of also being Dr. Soepriatna's PhD advisor. So Arvin is originally from Indonesia and grew up primarily in Texas before going to Washington University in St. Louis, where he majored in biomedical engineering and minored in electrical engineering. He graduated cum laude in spring of 2014 before coming to Purdue University, where he immediately joined my group, and ever since fall of 2014, became an integral member of the lab, doing a lot of different projects, but primarily focused on cardiac imaging, did a lot of different aspects of characterization of mouse hearts in relation to infarction specifically, and I'll get into that as part of his webinar here. But Arvin was just one of the great people in the lab and an outstanding scientist. He graduated in December of 2019 and is now, in the last few months, joined Kareem Kalom's lab at Brown University. So he's been there for a few months now. Certainly the world is in a different place than we were in January. And so things have been kind of turned upside down, but I'm thrilled that he's going to have a chance to talk about his PhD research from West Lafayette and where he's headed in the future, especially utilizing the Visual Sonics Vivo systems for characterizing cardiac dysfunction and disease in different murine models. And so with that, Arvin, I'll turn it over to you. Excited to see how you present, and I'm happy to answer any questions with Dr. Soepriatna at the very end. Thanks, Arvin.
Thank you very much, Craig, for the kind introduction. As Craig mentioned, my name is Arvin Soepriatna, and today I first wanted to thank everyone for all the participants for being here today to attend this webinar, and I hope everyone and their family remain safe during this pandemic. I first want to preface with the fact that the work that I will be presenting today covers the research that I did during my PhD at Purdue, as well as some offline Vivo 3100 work that I've done in collaboration with Visual Sonics. Today's talk will discuss how we can use four-dimensional ultrasound to non-invasively quantify myocardial infarct size in mice with two different approaches. As you can see on the left video here, this mouse has a pretty well-developed apical infarct with the infarct region outlined in dashed yellow lines. Using the 3D strain algorithm that we've established in our lab, we can quantify myocardial wall mechanics and generate these informative 3D strain maps to inform us of infarct location and kinematics. We can also use geometry information, particularly by taking the advantage of the fact that the infarct undergoes significant wall thinning to segment infarct tissue from 4D ultrasound data with the Vivo Lab software. The outline of this presentation will begin with a brief pathophysiology of myocardial infarction. I will specifically be focusing on infarct size and how it determines remodeling outcome. And then I'll talk about the advantages of four-dimensional ultrasound imaging and why we wanted to move from the 2D realm to 4D in terms of infarct characterization. And then I'll talk into more detail about the two methods I mentioned previously, the 3D strain approach, as well as the geometry approach using the Vivo 3100. I will then wrap up with the conclusion and future directions. As many of the audience members here are probably very familiar with, coronary artery disease remains to be one of the leading causes of death in the world. coronary artery disease is the progressive accumulation of atherosclerotic plaque in the coronary vasculature. What happens as the disease progresses is that these plaques often rupture, leading to clot formation and occlusion of blood flow to regions of the heart. And when this happens, it's really important for physicians to come in and open the occluded vessel either via stenting or angioplasty to reperfuse and allow blood to flow back to the damaged tissue. If this isn't done in a timely manner, then you get a significant damage in the heart, and the rest of the heart that is still viable and contracting will have to compensate for this loss of function in the damaged region. And this continual remodeling process to compensate for that loss in function is the basis of post-infarction remodeling. So what happens to the heart following a myocardial infarction? Well, immediately after an acute event, the heart undergoes this complex series of inflammatory and cellular processes. Specifically, early in remodeling, fibrotic scar tissues begin to deposit in the infarcted tissue. And this is a critical step because it provides some sort of mechanical rigidity, so you don't run into issues about left ventricular rupture. However, until the scar begins to fully develop and deposit, these regions are in fact very susceptible in undergoing infarct zone thinning and expansion. And this is a process that happens because the now akinetic necrotic tissues is constantly being pulled by the contractile myocardium, and this leads to the infarct expansion process. Depending on the size of the infarct, you may get significant ventricular dilation and hypertrophy, and these all are contributing factors that may lead to heart failure. When someone has a heart attack, there are two outcomes typically. One of them is fibrotic scars begin to develop early with enough strength and good mechanical properties, and then when this happens, you get an adequately healed MI, which results in preserved geometry. However, in some cases, you may have infarct sizes or they are pretty big, and this is because the collagen deposition and the scar formation, the quality of the infarct scar itself may not be very strong, and this may lead to significant infarct expansion process, leading to severe dilation and eventually heart failure. In fact, approximately, statistics show that 20% of MI patients tend to develop heart failure within 5 years of having a heart attack. And that kind of highlights kind of the importance of infarct size and remodeling outcomes. And then in preclinical settings, when people quantify infarct size, they still typically rely on two methods, the first one being histological evaluation. But this is a process that requires sacrificing the animal, harvesting the heart, and it's more kind of like an end time point. So if you want to study infarct expansion or disease progression, it's really difficult. You have to kind of stagger the study between different mice and sack at different time points. And we know from the biology of the coronary vasculature in mice that even if you do the same MI surgery, due to differences in coronary vasculature in mice, even within same species, you may get very different kind of infarct location as well as remodeling patterns. And this is why we wanted to evaluate a new way that doesn't require a terminal time point to quantify infarct size. Another commonly used method that enables longitudinal study of infarct sizing is the late gadolinium enhanced MRI. So this basically, you introduce gadolinium contrast agent into the animal, and due to the different perfusion state of the myocardial tissue, depending if it's healthy or if it's infarcted, you get this kind of later washout period in the infarcted tissue. And you can in fact see here in the infarcted tissue, you see whitening of the contrast agent. But this is a process that still requires a lot of animal preparation, and it still involves the injection of contrast agent. So with four-dimensional ultrasound, what we wanted to do is develop a non-invasive and label-free method to quantify infarct size throughout post-MI remodeling. And the system that we used for our study is a combination of the 2100 and 3100 system. All of the strain work that I will be showing in the first half of the presentation, we used the Vivo 2100 system. This was because the study actually started before the 3100 came into available. And basically, the 3100 is now a click button that allows and enables the four-dimensional acquisition, whereas in the Vivo 2100, we have a lot of custom MATLAB scripts that control the motor, and we also have to post-process and reconstruct the data after, whereas the Vivo 3100 does that automatically with a click of a button. One of the advantages of the 3100 or both ultrasound system is that they have this really high frequency transducer giving us axial lateral elevation resolution that allows us to kind of characterize and identify features in the myocardium regarding the contraction and also infarct location. One of the advantages though with the 3100 is that unlike 2100 with the single gating scheme, the 3100 has a dual gating scheme. What this means is that the image acquisition when you try to do gating is much faster. So it really speeds up the acquisition time in terms of minutes for one video at a particular slice to matter of seconds with the 3100. The way we acquired the 4-dimensional ultrasound data is we first anesthetize the mouse, place them onto a heated stage to make sure that they don't lose body heat. And then we attach the pods onto these gold-plated electrodes that allow us to acquire ECG and respiration waveform. We then, using the motor that's provided by the Vivo imaging stage, can move the transducer and acquire data in short axis mode from the apex all the way to the base. And this allows us to get an entire kind of volumetric data at different slices that you can stitch together. And then we can use the ECG and respiration waveform to kind of match everything across time points to get 4-dimensional data. And what I'm showing here is a representative 4D ultrasound data from a healthy heart. I wanted to showcase that even though we acquired the data in short axis, we can get these coronal plane and sagittal plane. And this enables us to kind of look at regional differences in terms of the contractile patterns. And you can then render them onto 3-dimensional volume to see how the heart contracts over time. And since that was a healthy heart, you see uniform contraction throughout. But the reason why 4-dimensional ultrasound is really important is to identify features in disease cases where you may have regional differences in contractility. There may be regions that are contracting more than others, or if you have uniqueness in geometry that cannot be appropriately captured using 2D approaches. So the model that we use for this study is the MI model and the permanent ligation model specifically. So in order to induce a heart attack, what we did is we first intubated the mouse endotracheally and connected it to a ventilator. We performed a left thoracotomy to expose the chest region and open the heart. Once we can visualize the left coronary artery, we then loop a suture underneath that vessel to induce a permanent ligation model of the infarct. And you can see here within 24 hours, this mouse developed a significant loss of function in the apical region here. Now, when you look at the 4-dimensional ultrasound data of the infarcted left ventricle, you can see regions of asymmetry in terms of contraction, whereas the septal wall here contracting a lot, the lateral wall don't really contract that much. And you see a similar pattern in terms of the apex compared to the base, where the base are contracting a lot, whereas those in the apex don't really. And then you can again render the 3D geometries using Vivo Lab to kind of get a sense of the kinematics and geometry throughout cardiac cycle. And now I wanted to talk about how we use those 4D data and integrated our own 3D strain approach to kind of estimate infarct size. And this is work that was recently published in the fall of 2019, in December actually, where we used 15 mice of 3 equal-sized group of 5 mice each. In the first group, we did a permanent ligation model to induce a large infarct. And the second group, we did a 30-minute ischemia reperfusion, and this involves kind of just 30-minute occlusion of the vessel, and then removing that PE tubing, and then cutting the suture so that we don't induce any more damage to the heart to allow the blood flow to go back onto the previously ischemic tissue. And then we also did a sham surgery where we looped a suture underneath the coronary artery in order to see if the actual procedure of looping the suture induces some sort of regional damage to the left ventricle. And then we used the Vivo 2100 ultrasound system to acquire 4-dimensional ultrasound data at the following time points here. And we also collected 4-chamber color Doppler and pulse wave Doppler across the mitral annulus to look at the early filling and the late filling of the left ventricle to quantify diastolic dysfunction. The ratio of the E and A flow wave here is typically used as a criteria to look at diastolic dysfunction. And then at the end of the 28-day period, we perfuse the heart with KCl to arrest in diastole before performing histological analysis for infarct sizing. And the reason why we wanted to do ischemia reperfusion is because this is more of a clinically relevant model. Most people, when they have a heart attack, they go to the ER and they get the occluded vessels opened up. And we decided to do 30 minutes because we wanted to induce different severity of infarcts, and we decided to do it the minimum amount to cause these non-transmural infarcts to see if we can identify infarct sizes for cases where there's non-transmurality just based on their kinematics with our strain approach. And the way the analysis workflow works is once we acquire the 4-dimensional data, we resample the data to isotropic voxels of 60 microns to make the analysis more simplified later on. And then I segmented the endocardial and epicardial boundary outline in red and blue. And we also segmented out the sternal artifact boundary. And the reason why we segmented this out is because these shadowing artifacts caused by the sternum leads to underestimation of strain because it kind of blocks kind of the field of view. And then independently from the segmentation, we pass the 4-dimensional ultrasound data to our strain algorithm. And I'm not going to talk into too much detail about how the strain algorithm works. I'll just give a brief overview. The strain technique that we decided to use is a 3D direct deformation estimation. Basically what we did, and this here is a representation of a 2D view of what was implemented in 3D. We basically divided the images of the 4D data onto grid systems, and at the center of each intersection grid point, define an analysis region. We wanted to see what is the appropriate warping function that allows us to kind of predict the deformation of this red box from one time point at diastole to a successive time point, either at systole or other phase in the cardiac cycle. We used an iterative optimization approach, looking at image intensity by minimizing some square residual errors. And by an iterative technique, we can get the best function or matrix that matches the deformation of that red box from one time point to the other. And because of that, we can get the 3D deformation gradient tensor, which we can get the Green-Lagrange strain. Once we have the strain fields, we use our segmentation to basically isolate the strain regions onto the myocardium to see how strain looks at the different depth of the tissue as well as different regions of the heart. And then we unwrap that myocardial strain profile onto a Bullseye map. And for those of you who are not familiar with the Bullseye map, the apex here is represented as the center of the Bullseye map, and then you can see how the strain profile changes as you go in a different direction, either anterior, posterior, septal, or lateral. And here are some representative data or ultrasound images of the 28-day period for the different animal groups. In the sham group, we noticed that our surgery didn't really cause any sort of damage as there's uniform contraction and healthy contractile profile over 28 days. On the contrary, in the ischemia reperfusion group, you begin to see this like localized region of damage in the apical and anterior wall of the left ventricle. And this is outlined in dashed yellow lines. And this remains relatively localized to a small region of the left ventricle, even throughout the whole 28-day period. However, this is not the case that you see with the permanent ligation group. In fact, you begin to see not only does the heart appear to grow in its volume and size, it also begins to dilate over time, which is a very different remodeling pattern than what we saw with the ischemia reperfusion. And then when we used the 4D data to assess function, we were surprised to actually see that these permanent ligation group were able to sustain LV volumes up to 3 times that of a sham group. And you can clearly see here the differences in terms of severity of the heart function as well. In the permanent ligation group, there's significant dilation, whereas there's minor, statistically significant dilation observed at the last day of the ischemia reperfusion group. And then the loss of function is you can see here as the immediate increase in peak systolic volume as soon as day 1. And so with this, we can show that the permanent ligation group underwent significant LV dilation and undergoes severe remodeling. And this is reflected as well when you look at ejection fraction to look at systolic and diastolic dysfunction with EA ratio. The permanent ligation group have a much lower ejection fraction than those in the ischemia reperfusion group, but both are statistically significant from the sham group. But then if we look at the Ischemia Reperfusion group, in terms of diastolic dysfunction, we actually don't see any diastolic dysfunction in that group. We only observed it, the E&A inversion in the permanent ligation group, suggesting that diastolic dysfunction is only present in this group, even though they both have systolic dysfunction. And now that we have the strain, we can overlay them onto the endocardial wall to see how the strain profile looks along at peak systole. And this, what I'm showing here is the geometries at maximum contraction, as well as the strain profile at that time point. The yellow color here represents high strain, whereas those in blue represent regions of low strain. In the sham group, as expected, based on the ultrasound data, we have very good contraction, so a field of yellow throughout the entire 28-day period. But when we look at the ischemia reperfusion, we were able to capture that loss of function by reduction in strain, in myocardial strain in the anterior apical wall. But when we begin to look at the permanent ligation group, you can see that regions that exhibited significant wall thinning also had much larger and lower strain values in that region as well, indicating a different remodeling pattern and different severity. And so we did two different ways to kind of quantify infarct size. One of them is solely from the geometry. We basically co-registered the strain maps and the ultrasound data, as well as our segmentation on top of each other. And then we reoriented the data such that we get the true center line axis of the left ventricle. We can then look at the geometry and then measure wall thickness throughout the entire perimeter of the myocardial wall. And then for regions that have thickness of the myocardial wall less than 0.5 millimeter during systole, we define that as a region of infarct tissue. And this threshold has been published in histological papers, as well as it's recently been used for ultrasound data as well. But now, since we have these strain profile, we wanted to see if there's more of a robust way to kind of quantify infarct size without too much intervariability due to the observer when you do segmentation. So based on the strain data, on the Bullseye map strain here, if we look at the strain profile as you go further away from the center of the infarct, we actually notice a sigmoidal behavior of the strain profile. And this is to be expected because in remote regions, we still have healthy contractions, and the infarcted tissue with the stiff collagen scar is going to have low strain values. However, previous studies haven't really reported the sigmoidal behavior because they haven't really been able to do true 3D mapping. Instead, they've just typically been showing that there's intermediate values of strain at the interface of the disease and healthy tissue. So we defined the inflection point of the sigmoidal profile to be the infarct extent. So we decided to do that at each 30-degree region and then try to reconstruct an infarct map based on the Bullseye map here based on our strain profile. And for regions that have significant sternal artifact, which we typically observe in the septal region, we couldn't rely on the estimation of this sigmoidal profile there. So we decided to interpolate in between reliable regions to kind of fill in the gap. And then we can then acquire these Bullseye strain map. And again, if you look at the baseline in sham, you see a healthy yellow regions throughout the left ventricle, whereas if you get to the ischemia reperfusion, we can quantify infarct size based on our strain profile. And in this region, you have regions of very low strain. I wanted to note that with our ischemia reperfusion group, we did not observe any, we did not observe transmural infarcts, so we were not able to quantify infarct size based on the wall thinning criteria. However, since the permanent ligation group did exhibit significant wall thinning, we were able to quantify strain infarct size using both techniques. And we get to see that there's pretty close estimation of infarct size between the two methods, and then the permanent ligation group have much greater regions of lower strain than ischemia reperfusion. If we look at the Bullseye strain map over the course of a 28-day period, we see expected trends for the sham group with healthy contraction throughout. And then in the ischemia reperfusion, we can highlight kind of the repeatability of the strain technique because we were able to reproduce approximately same location infarct throughout the disease progression. Whereas in the permanent ligation group, you can identify that there's significant LV dilation as the radius of this bullseye map gets bigger as you go over the 28-day period. And one thing that I do want to note is the strain estimated infarct size shown in black can kind of predict early on how big the infarct is going to be. If you look at the wall thinning approach, you have a relatively smaller region of this infarct size based on that criteria, but over time it continues to converge until it matches those or converge with the boundary of the strain estimated infarct size. And we believe this is because wall thinning is a process that happens, that takes a couple days to fully occur. There's a lot of resorption of tissues, so you don't really observe this wall thinning phase in mice until approximately 5 days into post-remodeling. And using this technique, we can actually quantify infarct size and monitor animal-specific disease progression. And it's really important, again, to use four-dimensional ultrasound because you notice here that there's different geometries in terms of the infarct size and location, even within the same surgical group. And again, this is because there's significant variability in the coronary vasculature of mice. And you see a similar trend here with the permanent ligation group, where some infarcts are more symmetrical than others, while some is only localized to the anterior wall and not to the posterior wall. And integrating animal-specific geometries with infarct sizing allow us to kind of better characterize disease progression in these models. And this is what I was mentioning earlier, where we were able to quantify infarct size based on our strain estimated approach using the for the Ischemia Reperfusion group. But we can't do the same thing with the wall thin approach because there was no transmural infarct. And then this is the convergence that I was referring to earlier. At the earlier time points, as the wall are slowly thinning out, you can see infarct size based on the wall thinning approach begins to kind of ramp up until it converges with those estimated early on from the strain estimated approach. And so this kind of suggests that using our strain estimated technique, we can kind of predict infarct boundaries in the early phase of remodeling that can be used to predict how severe the infarct is going to grow at the end of the remodeling process. And if we look at the strain profiles, we were able to find kind of some more interesting finding. In the baseline and healthy groups, you see healthy strain throughout the left ventricle. However, as you progress through the 28-day period in the Ischemia Reperfusion group, we find it interesting that we have significantly higher strain in the infarcted tissue. And this may be due to the ability, the fact that it's not a transmural infarct and there's more viable tissues there contributing to the contractile profile pattern. And this kind of also showed that maybe if you have more viable tissues, animals with less remodeling typically have a more contractile infarct scar, suggesting more viable tissue. And we wanted to compare how our infarct size compared to histological data. So we did collagen quantification to kind of get a better sense of how the strain profile looked and explain the sigmoidal behavior of it. As well as for infarct sizing, we decided to do a midline length measurements where you kind of draw a circumference. And then for regions that have significant wall thinning and significant presence of collagen, as shown here in blue, we define that as an infarct size. We then stitched together different slices of histology from different regions of the left ventricle, summed them up, and calculate infarct size as a percent of the left ventricle. And the findings that we observed from our histological data really support our strain data. If you look at the ischemia perfusion, we are indeed getting significantly higher strain values in the infarcted tissue when compared to the permanent ligation because there are viable tissues there that are contributing to the contractile pattern. Whereas in the permanent ligation group, you have entirely collagen wall scar in the apex, which means that they have very low strain. And if you look at the collagen content, this kind of helped explain the sigmoidal profile that we observe. Intermediate regions between the base and the infarcted tissue exhibit intermediate values of collagen. And then the infarct tissue for diseased permanent ligation and ischemia reperfusion showed statistically significant quantity of differences in terms of collagen content. And so we correlated the infarct size using our strain estimated technique and the wall thin approach with histological data. And we found that we have a better correlation with our strain estimated technique. when we look at how strain estimated infarct size compares to in terms of cardiac function, we also saw a better correlation with our strain approach when compared to the wall thinning approximation. And so we wanted to show what we can do with the Vivo 3100 in terms of infarct sizing. And this is primarily by taking advantage of the fact that infarcted tissues tend to undergo significant wall thinning later on in terms of disease progression. And the way we did it with the 3100 system is we used the Vivo Lab software, and then we opened this analysis tool, and we can segment out the endocardial and epicardial boundary, shown in red and blue here. So you wanted to segment them at both diastole and systole. And at systole, you freeze the screenshot, and then try to look at regions that have wall thinning or begin to exhibit wall thinning. And this is a representative slice just at the beginning of the infarct location. And this is where the wall starts to thin out and exhibit that thin wall criteria that's less than 0.5 millimeter in terms of distance. So you can quantify this thickness using the caliper tool down here in the Vivo Lab system. And then you iteratively go through that step from the base all the way to the apex of the left ventricle, and then you can render the data onto a 3D volumetric loop to kind of quantify these and identify regions of the infarcted tissue using the Vivo 3100 software. And this again highlights the importance of 4D because these infarcts are indeed not symmetrical. They're more located in the anterior and lateral wall as opposed to the septal and the posterior wall. And so we quantified function and observed significant differences between the MI model and the healthy case. They have much more greater volume of chamber volume in the infarcted tissue, as well as lower ejection fraction. And then we can quantify infarct size by simply looking at the volume of the infarcted tissue divided by the volume of the myocardium, which is essentially the volume of the epicardium surface divided by the endocardial segmentation. And then since we are using a wall thickness approach, we wanted to see what we can also do with the Vivo 3100 to look at diseases that induce differences in wall thickening. So this is a model of aortic constriction, which is a model of pressure overload hypertrophy, allowing you to get these thick wall. And I believe this data is from more of an earlier hypertrophy. So we did still see significant differences in terms of the wall thickness between the healthy and the TAC model, which resulted in a greater wall volume suggesting hypertrophy. So what are some takeaway messages of this talk? We have shown that by integrating 4-dimensional ultrasound with a 3D strain algorithm, we enabled essentially the in vivo quantification of infarct size. Having the ability to look at animal-specific LV geometries, and in the 3D strain maps, we were able to individually monitor disease progression and capture unique remodeling patterns based on the severity of the disease. And using this technique, we were also able to kind of quantify infarct size early on and predict to what extent the severity of the heart damage is going to be based on the strain profiles. And then for the wall thinning approach, we were still able to quantify infarct size, but we think it's more appropriate to use this approach for more mature infarcts and later time point of the remodeling process, as there's significant wall thinning in that region, and it makes it easier to kind of identify where are the infarcted wall based on the thickness. And so what is the eventual impact of this work? It is by having the ability to predict for final infarct size early on, we can potentially identify patients who are at high risk of developing heart failure following an ischemic event early on, which means that the doctors and physicians could provide more patient-specific treatment and better improve patient prognosis and quality of life. And with that, I would like to acknowledge everyone in the Cardiovascular Imaging Research Lab, especially my research advisor, Dr. Craig Gorgon. Without his help, I would not have been able to perform any of this work. And I would really want to I really appreciate his mentorship because he enabled me to succeed as a researcher. And I also wanted to thank the 2 undergrads that helped a lot with this project, Abigail and Kevin, as well as our collaborators who helped with the validation and creation of the 3D strain software or 3D strain algorithm, as well as the people at Visual Sonics for making this work be possible. And with that, I want to thank all of you guys for listening, and feel free to shoot me an email if you have any question about this presentation, and we'll go on to the question section of the talk.
Fantastic presentation, Arvin. Thank you so much. For our attendees, just as a reminder, if you have a question, if you can please submit that through the Q&A button located at the bottom of the screen, as opposed to putting it into the Zoom webinar chat box, that would be greatly appreciated. So we have quite a few questions that are pouring in right now. Arvin, so I know you said early on that you did the analysis for 4D on the short axis, but can this actually be done on the long axis, or do you have the potential to maybe acquire the image in a long axis, which would be a little shorter time frame for acquisition, and then rotate the image and do the analysis on the short axis?
Right. So the nice thing with the 4-dimensional acquisition is, regardless of the plane at which you acquire the images, you can get images in other planes as well, which is why when we are looking at the Vivo Lab kind of the 4 panels, even though we collected the data in short axis, we were able to see pretty good resolution data in the long axis view. So yes, you can acquire in long axis. The reason we did it with the short axis view is so that we want to better capture kind of like the radial contraction pattern in this beating part.
Fantastic, thank you. Can you comment about what are the average heart rates during capture and the time that it takes to actually acquire each 4D?
Right, so average heart rate for these mice range from like 450 to 600 beats per minute. So the acquisition time differs depending on whether you use the Vivo 2100 system or the 3100 system. The way we did it with the 2100 is, like I mentioned, we have a bunch of custom MATLAB script that control the position of the transducer. And because it doesn't have the dual gating capability, it takes approximately 45 seconds to a minute to acquire one video of one slice. And we typically acquire around 40, 50 slices to get the entire volume of the heart. So with the 2100 system, we typically take 40 minutes to acquire one 4D data set. But I believe with the 3100, we can get this in 10 minutes at most.
Yeah, thank you. So just to add some more information to that answer, to acquire a 4D image on the Vivo 3100, it is dependent upon the parameters that you set during acquisition. So the 10 minutes that Arvin is referring to is actually the maximum amount for each possible setting, including the smallest possible step size. So if you were looking to do a very rapid acquisition, the acquisition time is closer to about 2 minutes. So moving on to the next question, it kind of builds off of that. How could you minimize the amount of work and improve the image quality when delineating the contours of the left ventricle during 4D analysis?
Right, so getting really good ultrasound images during acquisition is key here. As you can tell, like from the presentation, I also segmented out the sternal artifacts. This is because these are actually really hard to avoid. The best way that I found is when we acquire the data, it's best to acquire at an angle from the left side of the chest of the mouse. That way you're trying to kind of get rid of as much of the sternal artifact from the field of view as possible. And if you get like better boundary detection, or like less artifact, then it's easier to segment as well. And I believe you can also use contrast agent. There's a contrast mode in ultrasound if you can get the contrast microbubbles from Visual Sonics as well.
Great, thank you. So we have a question about the threshold of the 0.5 millimeter wall thickness to define the MR region. One of the attendees would like to know if it's possible to use any portion of the wall in which the thickness was lower than the baseline, so the pre-MI scan.
So another way that people have done this is by looking at percentage of thickness based on their healthy region. You can do that as well. it just depends on what threshold you decided to use. We use that cutoff because we found a publication that uses it and want it to be as consistent as possible.
Great, thank you. So I have a follow-up question asking specifically about a model of atherosclerosis. I know that that isn't necessarily something that you did in this study, but I know that your lab has experience with AAA models using the 4D to image abdominal aorta. So the question asks, are infarcts actually observed in LDL receptor knockout mice in atherosclerosis?
That is not a question that I know off the top of my head. I do believe there is a double knockout model with the LDLR and some sort of, I'm not sure, it's maybe a combination of that and ApoE that tends to develop spontaneous infarcts, but I can't really comment on how often and at what time point they begin to develop infarcts. But if you have the infarct, we could probably quantify it using the tools that we've developed.
Hey, Arvin, this is Craig. Let me jump in just real quick to say that mice are really very resilient, and the chances of finding a heart attack, even in the hyperlipidemic LDL receptor knockout or ApoE knockout mice, is really fairly low, even if you put them on a high-fat diet. So the field that uses those hyperlipidemic models, my understanding is they almost always then combine it with some type of coronary ischemia model and or hypoxia model to induce heart attacks and better mimic what it would be like in a clinical setting where you'd have both a ischemia damage and atherosclerosis. But just LDL receptor knockout mice themselves, in my opinion, understanding, they don't develop heart attacks at any regular frequency for studies.
Great, thank you, Craig. So the next question is, how do you select the regions of the heart used for your histology calculations?
So we acquired a lot of slices from our histological data. I think we embedded the whole heart onto a paraffin, and then we sliced at different slices, and then acquired approximately 5 to 10 regions of the heart and kind of stitch those together based on the infarct sizes that we get at particular slices. So we did acquire several slices across the thickness or the entire left ventricle so that we can get a better accurate representation of what the infarct size is from histology. Does that answer the question? Okay.
Yes, thank you. So the next question is, in your estimation, how does 3D, 4D strain algorithm shown here compare or correlate to that in the 2D measurements?
So we did not really do a comparison with the 2D because it also depends on how you can, the 2D strain, we didn't, are we talking about the Vivo strain software or?
They didn't specify, but I would assume that's how most people are actually calculating strain off 2D images.
Mm-hmm. So we don't have the Vivo strain software in the lab, so I can't really comment on that. But I think with the Vivo 2D strain, because it's a 2D view of the left ventricle, you may be missing regions of the heart that are out of plane that actually have these low and regional differences in strain profiles. I think within the plane, you can really identify regions with low strain, but we wanted to see how the strain profile kind of behave in 3 dimensions, right? Because the heart really contracts in 3 different directions. It twists and undergoes rotational motion on twisting and like wringing a wet towel, right? So we wanted to see the different components of the strain as well.
Hey, Arvin, can you bring up the strain slide where we talk a little bit about the methods with the DDE approach?
Yeah.
So when I read this question, it made me think about trying to just clarify a little bit more about what we're showing with strain versus what the typical global longitudinal strain, global radial strain, global circumferential strain, clinical assessments have typically used. And so, right, our thought is that we can use these data and absolutely compare it to the GLS metric that's been common in the clinic. But strain itself is a 3-dimensional tensor. It's not a number. And so when we look at all these different directions, you really need three-dimensional and four-dimensional data to understand what's happening, not just in the one plane you're looking at, but in all of these directions, in three directions, and in the shear components as well. So that's why we thought it was a great use of this DDE approach to apply it to this four-dimensional ultrasound data, where you can use the speckle pattern throughout the myocardium to calculate the full three-dimensional Green-Lagrange strain tensor. And then from there, we kind of skipped over some of these methods, but what we did was use the first maximum principal component. And Arvin's basically showing the magnitude of that principal component without directionality, just to get a rough estimate of where is strain high and where is strain low. But that doesn't mean we can't go back and look at longitudinal strain and create a GLS-like metric at each of these slices for comparison to 2D data or other studies. But in general, where we see high first principal component Green-Lagrange strain, we're going to find high global longitudinal and global radial strain numbers as well, and vice versa with low strain values for global strain and green strain. Anything to add to that, Arvin?
I think you summed it up perfectly.
Great, thank you, both of you. We have a question about the isoflurane concentrations and how that might affect heart rate. Can you just comment about any difference in iso concentrations that you used?
Right, so there was concern when we first started the study if we use a standard tabletop vaporizer, because heart function really is a function of the anesthesia level of the mass as well. So what we did is we use a low flow flow vaporizer, tabletop vaporizer called the Somno Suite. And so we were able to deliver much lower amount of isoflurane levels in a way, even though we used kind of a 3% that we slowly somewhat taper off throughout the imaging acquisition to try to maintain a constant heart rate throughout the 30-minute of imaging. But using a tabletop low flow vaporizer really helped us get consistent kind of heart rate throughout imaging acquisition.
Great, thank you. Arvin, you're receiving a lot of praise through the Q&A. Many people are commenting that it's a fantastic talk, very interesting presentation.
Thank you.
So just going back to some more questions here, do you think that a longer ischemia reperfusion time, let's say maybe 60 minutes of ischemia, would allow to identify infarct sizes with your approach?
Yes, I think that with the longer ischemia reperfusion period, or longer ischemia period before reperfusion, we definitely would be getting a lot larger infarct sizes. I think if I go back to the slide that showed a paper, let's see, give me 1 second. I believe it's over here. Yes. If you look at this slide, even within an hour, you get to see this transmural infarct. And so you could probably look at the wall thinning approach as well, in addition to the strain. But the nice thing with the 30-minute ischemia reperfusion is using our strain software, or using the strain algorithm that we have, we can identify within the thickness of the myocardial wall how much of which the epicardial wall has lower strain than the endocardial wall, let's say. So we have thickness strain mapping as well.
Great. Arvin, can you comment about the Simpson's protocol and how that might compare to a 3D, 4D acquisition of the heart?
Right, so the very first publication and study that I did was actually comparing 2D approaches for LV function with 4D approaches. And then if you, I believe I have a slide on that. Let's see here. This is from my first publication. And you can tell that depending on the imaging plane that you acquire the data in, you may be able to, you may miss some of the regions that are more akinetic. So you have this larger variability in terms of what you get if you use the Simpson's approach when you compare it to the direct volumetric approach from the segmentation of the four-dimensional data. So it just allows for more reliable and consistent measurement without worrying that you're acquiring images being out of plane or not in the correct plane, missing a little bit of the infarct and such.
Great, thank you. We do have quite a few questions that are still coming in. In the interest of time though, Arvin, I'm just gonna narrow it down to kind of 2 more, but we will be happy to email these questions over to our presenters, and they said they would be happy to email you back with a response. So does obtaining the short axis slices at inconsistent levels during different time points change the precision or accuracy of the quantification of the infarct?
Sorry, can you repeat the question?
Sure. So they're asking if obtaining the short axis slices at inconsistent levels, so let's say you're imaging on day 1 and then again on day 28, it would be hard to get the exact same 2D slice, even though you're probably using the exact same step sizes and things of that nature. So they're wondering how much that changes the precision or accuracy of the quantification of the infarct zone.
So in between imaging period, we always use kind of the same, again, step size with image width. And then when we try to compare the strain data, we actually reorient the data so that in terms of their mitral valve and in terms of the apex, they're always aligned between animal data set. So when we want to look at the, let's say the Bullseye strain mapping, we are quite sure that they are all registered in terms of the same orientation. I guess the worry is like you worried if there's a shift, right, in terms of if you're not orienting it exactly the same way each time, then the Bullseye strain map might be slightly rotated in some than the other. But we did use, we did match them all to the same kind of coordinate based on their physical characteristics.
And I'll add too that we put out a tomography paper in 2017 as well, where we did a comparison where I think it was a 40 micron step size, and we segmented every single slice from apex, below the apex, to above the base, and then we threw out every other slice and every third slice and every fourth slice, and gotten a sense of how many slices are actually needed to create a robust segmentation, and found that up until about 200 microns, you can get good segmentations with a 200 micron step size pretty reliably. So you shouldn't think about doing acquisitions where you are necessarily doing the 50 micron steps if you don't necessarily need that. That's going to take a lot longer, and you probably won't actually use that data. You use every other or every third slice when you're doing the segmentations. So we're definitely not having to go back and find the exact same location each time with this volumetric approach. It absolutely helps with repeatability, where we can go back and collect a whole new volume. And even if it's at a slightly different angle and a slightly different starting point or ending point, we can get this similar data and similar results from it.
Yeah, and it also brings an interesting point, like the trade-off between how much data you want to collect and the resolution of your final strain. So what I did is I kind of used the limit of the minimum elevation resolution of the transducer to kind of guide that as my step size. But you don't really need that fine detail if you want a coarser estimation of strain. We just wanted to get as high resolution as we can, so that's why our acquisition time is also longer, because we wanted to get as much data as we can from these ultrasound data.
Great. Yeah, thank you, Craig, for pointing out that earlier paper from your lab as well. That's a great tool to use in terms of cutting down the acquisition time as well. So we have several questions about translating this kind of 4D imaging approach and 4D strain approach to the clinic. So to kind of summarize all of them, I'll just ask, in your opinion, how easy is this to translate this technology to humans?
I think the most challenging is the acquisition of the 4-dimensional data in humans. Yes, we do have 4-dimensional probe now, but it doesn't acquire the full volume of the left ventricle. It can give you data on, let's say, the valve motion, but I think the way we acquired the 4D data is like we require respiration and ECG gating. That means that most people don't get their respiration waveform or a full kind of ECG map with the 12th lead to try to match them in between slices as well. And there's also an issue of hardware of wanting to kind of, how do you reliably kind of acquire consistent spacing between slices. And I think the limitation is more on the hardware component, because the software is, once we have the data in 4D, we will need to change some parameter of the strain code so that we can take into account what the correct analysis region is in terms of averaging kind of blocks of region to look at strain. But I think those are smaller hurdles than the actual hardware component in terms of acquisition.
And yeah, I mean, I think as the technology advances, especially with the 2D arrays, we're going to be getting to the point where it's not just focused on aortic valve and mitral leaflet dynamics, but ventricular contraction with these volumetric assessments. But I think the first generation is likely going to be acquiring multiple volumes through different windows, through different cardiac windows in clinical and large animal studies, and then collecting and merging those volumes together, those 4D data together, similar like we're doing slice by slice with the small animal approach, to get clinical data that is similar to what we see here with the mouse.
Well, I just want to take this time to thank you, Arvin, for this fantastic presentation today. And of course, thank you, Dr. Gorjan, for your time as well. I would like to remind all of our attendees to please visit our Visual Sonics website. All of our webinars, again, are recorded and can be found on that website, as well as a variety of other resources for you, such as image galleries, publications. There's also a variety of protocols and things. And by all means, if you're interested in learning more about the 4D technology on the 3100 or how to do this analysis, please reach out to our application scientist team, who would be more than happy to help you with this. So again, to our presenters, I thank you both for your time today, and have a fantastic rest of the day.
Thank you,
Sarah. Thanks so much for the opportunity to present. We appreciate it. Thanks for everybody participating and giving such great questions.